that's all acids and bases. So in chapter 7, it's really talking a lot about stuff we've talked about, but just trying to make sure we put things together. Okay, so they call it what's the attraction? Well, it's all about how molecules interact. So the things we're going to talk about tonight is we're going to try and get through attractive forces, the characteristics of water, which wouldn't be if you've had AMP. That's not something that's brand new because you talk about the universal solvent all the time. And then also like what are the rules for dissolving molecules, mixing molecules. So when we're talking about these, they call these intermolecular forces. So the term inter means between, molecular forces. So we are not talking about what holds two atoms together. We're not talking about what makes a molecule. We're talking about what holds molecules together. Why do molecules like to cluster around each other? Why do they form liquids? Why do they form solids? So they cover it from weakest to strongest. So the weakest of all of the forces is called London forces. Some books call them dispersion forces. That's another example. London forces are the only attraction you see with nonpolar molecules. So nonpolar molecules, they only have London forces. These are the weakest of all of the forces. They create these, like they say, instantaneous attractions. The best example, and I do kind of like this example, is like what happens with saran wrap. Right? So you get saran wrap and you peel it off and then you tear it, and then what does it do? Yeah, it like folds back on itself, and then you go and try to pull that corner, and then another one folds, and then pretty soon the middle is folded in half, and then you get so irritated you throw it away. Okay, so that's kind of like London forces. So that seems like a random thing that happens. Well, occasionally, long, nonpolar hydrocarbons, like fuels, like octane, like butane, propane. So these molecules, they share their electrons evenly. Remember that they make like a really neutral molecule, but occasionally more electrons are on one side of the molecule than the other. And so you can see that in this one. So when that happens, can you see how if the electrons are more towards the top, that would create a slightly negative region, less at the bottom, slightly positive. So that means if this happens between two molecules that are close together, then there may be this attraction. But as soon as the attraction occurs, the attraction actually disappears. So the electrons, because remember electrons are moving back and forth between carbons and hydrogens. So sometimes they may end up more on one side than on another side. And you might get this London force. But it's pretty weak. And the best example is when we did lab with the cleaning. And I took hexane. Hexane's a great example. In fact, this is heptane, this one that they have here. But with hexane, I put some drops on the countertop. And remember, it spread flat. It didn't bulb, it didn't form a bubble, it didn't clump, it just like spread out, and then as you watched it, it just evaporated. And so it was gone pretty quick. That's because this is the only thing holding molecules together. So this is really weak, if I could spell. So it spread out on the lab bench. So it just formed like this really flat puddle. Not a lot of attraction between molecules, so it doesn't really form. There's no drops that it forms. It doesn't cluster together. And it evaporated really fast. It was the fastest of all three of the ones. We used water, we used isopropyl, and we used hexane. So this is why small fuels like methane, propane, butane, they're all gases at room temperature. They're all bigger than water. Size-wise, you would think that they would want to like cling together and hang out because they're bigger, even to the point of hexane, 6 carbons, a lot bigger than water, but still evaporates really fast. And it's because there's nothing holding these molecules together. London forces, and those are super weak. So non-polar molecules have London forces, but they're super weak forces, and that's going to mean that they tend not to have any attraction. But ethyl acetate, one of the cleaners, the one that smelled like nail polish remover, ethyl acetate has that oxygen. And remember that we said if you see a nitrogen, oxygen, fluorine, or a chlorine in a small molecule, these 4 atoms are all what? They're the electronegative. So these are the electronegative atoms. And if you only have one, they are going to pull electrons towards them, creating this slight negativeness. So this side becomes more positive. That slight negative, slight positive, they call it dipole-dipole. It's really just the characteristic that polar molecules like to hang out. They like to stick together. So polar molecules also have London forces, but they really don't affect them. They're the weaker, so this is the stronger force. Polar molecules with one nitrogen, oxygen, fluorine, or chlorine are going to have an unequal sharing and create this dipole-dipole. So if another ethyl acetate molecule comes along, the slightly positive and the slightly negative will have an attraction. So these molecules tend to cling together more. They tend to stay liquid at room temperature. There's more attraction between the molecules than the nonpolar. But there's one that's even stronger. These are polar molecules where the nitrogen, oxygen, or fluorine has a hydrogen. If there is a nitrogen, oxygen, or fluorine with a hydrogen, this is polar, but if another molecule comes along that also has an electronegative atom, instead of just having this slight positive, slight negative, there is actually an interaction that occurs between the nitrogen of one molecule and a hydrogen of another molecule. And that's where the name hydrogen bonding came. Hydrogen bonding occurs when you have molecules that have nitrogen and oxygen, fluorine, but you don't see that in human, like you don't see that in living things. Pretty much it's just nitrogen and oxygen. If they have a hydrogen on the nitrogen or oxygen, then it can create this. And this is twice as strong as just dipole-dipole. This creates this attraction so that these two molecules tend to want to stay together. Instead of just like kind of being a weak attraction, this is a stronger attraction than just being polar, because they actually almost make like a semi-bond. Now they make almost like they're like a big molecule. Instead of two single nitrogen molecules, it's really like it's one nitrogen molecule. One molecule, multiple nitrogens that are sharing their hydrogens. The best example, and this one shows, like the first one here, this one shows how hydrogen bonding, so here's like an alcohol group. An alcohol group actually can like double back and interact with an oxygen. This middle one shows what happens though if you have acetone in water. So acetone has that electronegative oxygen, and the two that are above, water molecules, that's the hydrogen bonding. So these will mix easily and stay mixed because of this ability to interact. Same thing here. This shows ethanol. So having that oxygen in ethanol, but that ethanol also has an oxygen with a hydrogen, so we can hydrogen bond with water molecules. Hydrogen bonding with other hydrogens of water molecules and really creates this bigger attraction. So water is really the big one. So water is really considered kind of like unique. It's the universal solvent. I'm sure you've heard of that if you've ever taken any kind of biology class, because we are 2/3 water. Water has an oxygen with 2 hydrogens. So it can hydrogen bond. Its 2 hydrogens can hydrogen bond with other things. And then it has those non-bonding electrons, so it can hydrogen bond with 2 other hydrogens of nearby molecules. So there's a whole lot of interaction that can occur, and it creates an attraction. They say that a hydrogen bond is about 1/10 the strength of a covalent bond. So it's not as strong as a covalent bond, but it does create this attraction so that they start lining, molecules start lining themselves up in order to interact. Hydrogens interact with nearby oxygens and nitrogens. Nitrogens align themselves to be close to hydrogens. And so instead of just having a water molecule, it's like if you put water, and remember water's got that bent arrangement because of the non-bonding electron. So it does create that bend to it. That's its sort of normal position or shape. So this side is the slightly negative. Over here, these become the slightly positive. So that's the polar, the dipole-dipole. But even more so, another water molecule comes along, and it will automatically arrange itself so this oxygen is close to a nearby hydrogen. And then another water molecule will come in, and it too will arrange itself so that it's close to another hydrogen. Well, this oxygen can come along, and it can interact with a hydrogen here, here, and here. So you can start to see interactions between oxygens and hydrogens. They just continue to sort of tuck themselves in. Same here. Hydrogen bonding, hydrogen bonding. So instead of this looking like 5 water molecules, it really starts to act like one big complex. One large mass of water, because even in this, so the little hash marks, I have 1, 2, 3, 4, 5, 6. So I have 6 hydrogen bonds. So we said 10 of them would be the same as saying these are covalent bonds. So the more hydrogen bonds you add, the stronger the attraction. So instead of being like 5 water molecules, water acts like a complex, like a large, like mega molecule. When you look at other molecules that don't hydrogen bond, they don't have near the attraction. Here's an example, H2S, hydrogen sulfide. So oxygen and sulfur are in the same column. They both form 2 covalent bonds. Hydrogen sulfide is bigger than water is, H2O, but it can't form any hydrogen bonds. It is polar. It's really weakly polar. It's not very strong, but it can't form those hydrogen bonds. And so look at its melting point. So hydrogen sulfide melts at -80, whereas water melts at 0. So water, because it has that clinginess, has a much higher melting point and boiling point compared to other molecules its size. Like methane, CH4, that's roughly the same size as water, and methane is a gas. So at room temperature, methane exists as a gas because it's really small. But water, because of hydrogen bonding, keeps those molecules really closely collected. So here's some of my examples. Water has this really high melting and boiling point for its size. Other molecules its size, if they don't hydrogen bond, they're a gas at room temperature. Water has this high specific heat, and we talked about specific heat in the very first chapter. Specific heat is a measure about how much energy a substance can absorb or release and change temperature. So remember the analogy of like the jungle, about like your Caribbean tropical areas versus the desert. So the tropical areas have a lot of moisture. So there's a lot of plant, there's a lot of vegetation, there's a lot of water, it's really humid. And what is the temperature like? In a 24-hour period in Cancun, the temperature goes from, it goes from like 80 to 85 and then cools off to 80 and then goes up to 85. So the temperature doesn't change very much because there's so much water. Lots of water means that the temperature changes really slowly. But in the desert, you don't have water. So in the desert, you have sand. Okay, so in the desert, what happens to those temperatures? Yeah, they go really high, really low. So in Arizona at night, it can get down into the 40s. And during the day, it can be 110. So you can have huge shifts because there's no water in that environment. And so it's water that helps to maintain constant temperatures. It's one of the benefits with us having 2/3 water. That means that our body temperature changes temperature relatively slow. So if you go outside and it's hot, that increase in temperature is much slower than it would be if you were made of any other kind of solvent liquid. Same thing with the issues of little kids and fevers. So what can happen to a little kid in terms of a fever? It can spike really fast. But you have to think about like infants and toddlers, size-wise, they're 1/3 to maybe 25% of your size. So do they have more or less water? Less. Well, they have more percent, but they have less body volume. And that's why, since they don't have as much water total, their body temperature can change faster. So that's why they tell you, like, if you have a kid and it's got like a 99, 100 degree temperature, so you don't need to give them anything, but you do need to watch it because they can go from that to 102 in like 20 minutes. And for you, you would be like shaking on the floor if that temperature change happened to you. It would really be very critical if it happened to an adult. But it can happen with kids because they don't have as much water volume to absorb that heat energy. Cohesiveness. So this stickiness. And so if you look, the raindrop, that's what that's supposed to be. This raindrop, the reason that water falls as a drop is because water molecules are collecting. So all those water molecules have this attraction. Remember that in lab, when you look at the graduated cylinder, the curve of the meniscus in the graduated cylinder, if it's anything glass, it'll make this curve. And that's because water is attracted to the charged nature of the glass, and it pulls it up the sides. Surface tension, another example of hydrogen bonding. So in that one, I drew 1, 2, 3, 4, 5. I only put in 6 water molecules underneath the little bug. Those 6 water molecules, though, create a semi-solid liquid. So it's almost like a gel on that surface, and that allows the little water bugs to be able to sit on top. They look like they're floating, but they spread their weight out because their legs stick out really far. And if you ever looked at them, you can see they create like a bend, a dimple in the surface of the water where their feet are, but it's not enough weight to break the hydrogen bonding. Has anybody ever done a belly flop? Have you ever water skied and fell? Okay, if you've ever seen anybody fall when they're water skiing, they look like they're skipping across the surface. That is because when you're moving at a high rate of speed across the surface tension is strong enough to actually hold you up. So that's how come you can ski across the surface or even skip a rock. All of those are because of surface tension. And then it's not funny though when like you fall and like you and you do like these tumbles, and it really feels like something solid is hitting you as you're bouncing across the surface. If you've ever done a belly flop, that's the same thing. You've broken a lot of hydrogen bonds in order to be able to go in the water. So one thing, like the professional divers, the whole thing is it's all about the splash. Like they do all of these flips and spins and everything else, but when they go into the water, they want to go into the water in exactly like a straight perpendicular position. They want to be bent. They don't want to be sideways because that'll break more hydrogen bonds and that'll create bigger splash. So if they can go in and like where their fingers go in is where their toes go in, that is when you get the least amount of splash, and they end up with better points because of it. And it's all just because of surface tension, trying to break that surface tension. So we're going to talk a little bit more about this mixing, because the thing to keep in mind is that if you have polar molecules, water can interact with them. If you have salts, water can interact with them. Because of the charged nature, water has the ability to dissolve many other things. But lipids are a challenge. And so that's kind of like the end of this chapter, is talking about lipids and how you try to get them to stay mixed, even though oil and water are going to separate out. So with the lipids, with your fats and oils, that's kind of the challenge. So London forces, weakest. Dipole, dipole, regular polar, next, stronger. Hydrogen bonding, stronger than those. Number 4 on the list is when you have salts and water. So when you have a salt, what in the world? When you have a salt and water, that will create this attraction. And this is the reason that salts can actually dissolve in water. So if you look at a water molecule, do you see how the oxygen of the water molecule has that slight negative charge? And you see that it has attraction then for the positively charged sodium ion. The hydrogen sides of water are going to be attracted to the chloride ions, because the chloride ions are negatively charged and the hydrogens are the more positive regions of the water molecule. So if you take a crystal of salt, which is what the what the green and purple is, that's just a crystal of salt. And so it forms a real regular crystalline arrangement. You drop salt in water. Immediately, the oxygens begin to surround the sodiums, and they can then pull it away from the rest of the crystal. The hydrogens of the water molecules begin to surround the chlorides. And so this just continues, and it's like you can like disassemble the crystal. So as long as water molecules can surround the ions, it allows them to dissolve completely. So the negative part, the polar part of the water molecule, can interact with sodium and can interact with the chloride ions in order to allow for the salt to dissolve. The strongest attraction, though, that's not covalent, the strongest attraction is really between ions, between charged particles. So when you have sodium and chlorine, the reason those stick together is because of this ionic attraction. So they're not bonded like covalent. Covalent, remember, is sharing, and so that's a real stable bond. Ionic, the charge is the reason that you end up with the attraction. So in molecules, especially things like proteins, and in the nucleic acids with DNA, you will see that there will be some regions that have a negative charge, and they are attracted to regions that have a positive charge. So this is this ionic, charged, opposites attract type of interaction. And it creates the strongest of the bonds. This will hold things together more than hydrogen bonding. This will hold things together more than just salts and water. So the ionic attraction is the strongest of the intermolecular. So when you go to look at them, looking at them, if you were given an example, so like in the first one, the thing to do is to figure out, okay, is it polar, is it nonpolar? Is it ionic? Is it a salt? Okay, so if it is nonpolar, then you're going to see nothing but carbons and hydrogens, right? So the hydrocarbons, this is propane, these are the fuels, and you know that these fuels, they don't mix with water, they're nonpolar. So if you see nothing but carbon and hydrogen, they're nonpolar. They're your fuels or oils. And the only type of attraction that they have that holds them together is what? What was the weakest? Those London forces. So you would say the only force that holds them together is just London forces, and they're not very strong. So the next one, you're looking at a nitrogen, oxygen, fluorine, or chlorine, and there's no hydrogen connected to it directly. If there's only one, then we say that that molecule is polar or non-polar? Polar. So if there's only one, then it's going to be polar. But notice in this, there's no hydrogen on the oxygen. So this would have just the simple polar interaction. And that polar interaction, they just call it dipole-dipole. So that's just like plain, it's a plain polar molecule. But with the next one, here, one oxygen, so just like the one above, it has a nitrogen, oxygen, fluorine, or chlorine. It only has one. So we know that it's polar because it's small and only has one nitrogen, oxygen, fluorine, or chlorine. This one's a little different because the oxygen has a hydrogen. So if the oxygen has a hydrogen connected to it, like covalently bond like an alcohol, now instead of just being polar, this is polar, but it can hydrogen bond. So this molecule is going to cling together more than the molecule above. So the next one I always tell people, I'm like, okay, if you see it looks like there's some kind of salt, which is a metal and a non-metal, some kind of salt in water, then you know if it's some kind of salt in water, that attraction is always called ion dipole. And that allows the potassium chloride to dissolve in water. So if you take the crystals of potassium chloride, add them to water, they'll mix completely. And it's because we're putting ionic into polar. Putting salts in water. They have the ability to interact, and they're going to stay mixed. They won't settle out. You won't see salts just collecting on the bottom. They'll stay mixed because of this interaction. But of all of them, the strongest, the highest interaction between molecules, what forms crystals of salt, is because you have metals and nonmetals. So when you talk about iron oxide, you don't just have a single iron oxide atom. You actually have lots and lots of iron oxides all linked together. So this is just a salt. If you see just a metal and a nonmetal, then it will form a large crystal. And that is because it's ionic. So we're looking at weakest to strongest. So if you have sodium chloride as a salt, another sodium chloride will automatically align itself so that sodiums will be close to chlorines, chlorines will be close to sodiums, and they'll just keep building, and they'll form into these very sort of geometric crystalline arrangements. And that's because of that ionic interaction. So you don't ever have like a single sodium chloride molecule. You always see sodium chloride as a crystalline structure made of thousands, even millions of sodium ions and chloride ions. And the only way to get them to break apart is put them in water. Because like if you take salt, you have to heat it to about 3,000 degrees before it liquefies, before you break the ionic interactions. That's how stable it is. So it tends to stay as this solid, unless you put it in water, because the water can interact and separate the little ions and they float all about. But salts by far are the strongest intermolecular force. So this rule, The golden rule of solubility with this is that like dissolves like, right? So polar things mix with polar, non-polar things mix with non-polar. And so when we were talking about polar and non-polar molecules, I was telling you that oftentimes you can like just think about, okay, well, what things mix with water? Those things tend to be polar or they're ionic. They're one or the other. Non-polar things, those non-polar things that they'll mix. But when you try to mix polar and non-polar, the polar molecules just want to interact with each other because they have that slight positive and negative chargedness to them. So there is this attraction, and they literally push non-polar molecules out of the way. So this is the issue that you have with salad dressing with oil and vinegar, is that if you mix them together and you shake them, you can disperse the oil and the vinegar together, but as soon as you stop shaking, what does it do? It separates out, right? So you like have to pour really fast. To shake it up, and then pour it really fast in order to be able to get like an even amount so that you don't just pour oil on your salad or just end up with vinegar in the bottom, because that happens if you don't actually like mix it, keeping it mixed as it goes through, because they're very unlike. They're oil and water are about as far apart as possible. Oil being non-polar, water wanting the hydrogen bond. They do not want to have any interaction with each other because of that difference. So there's vitamins that have chemical structures that make them polar or non-polar. So instead of calling them the polar vitamins, they call them what? What are the two categories of vitamins? There's the B and C, and there's the A, D, E, and K. Soluble? What soluble, though? They call them one or the other. So the B's and C's they call water soluble. The water soluble vitamins, are they going to be polar or non-polar? They're going to be polar, just like water. So being polar means that you're going to see oxygens either irregularly arranged around. So looking at these two, one is polar, one is non-polar. Which one do you think is polar? Nitrogen, oxygen, fluorine, or chlorine, and you want an uneven arrangement. And remember that all these little corners, these are all carbons. So vitamin C or vitamin E? Vitamin C. Okay, so vitamin C has those alcohols. Remember, like the alcohols in your sugars? That's the reason why sucrose, fructose, glucose, why they're soluble in water. Those alcohols improve their ability to interact with water, so they're soluble. So this is polar. In fact, it can hydrogen bond. So you find water-soluble vitamins are found in more water-soluble environments. So these are all your vitamin C and all of the B's. Now what's interesting with this is since these are water-soluble, if you ingest a lot of them, they dissolve in your blood and your kidneys just flush them out. So when you're sick, what do you oftentimes drink to try and get better? Orange juice. So you go out and you buy like a half-gallon orange juice and like you just keep swigging orange juice. Well, vitamin C is ascorbic acid. That is the vitamin that's found in orange juice. And what it tends to do is it actually helps to stimulate your immune system. So that is the idea behind it, is taking this vitamin C. It also promotes repair. So there was a thought along the way that if you took a lot of vitamin C, that it would actually help to repair damage that occurs in the body with aging. So there were some people, you only need to have like 30 milligrams of vitamin C a day. You don't have to have very much. But there are some people that'll take 500, even 1,000 milligrams. So you only need like 30. They'll take 500. So they're taking more 10, 25 times what they should. So overdose of the water soluble, is this going to hurt you? No. It just gets excreted. Because it dissolves in the blood, it's carried through the watery environment of the blood, so excess just gets excreted. Vitamin E. See the long carbon chain? The non-polar look? Remember, like our fatty acids had that long carbon chain, the zigzag sticking off? So those big non-polar areas, instead of water soluble, they call these what? Lipid or fat soluble. Okay? Fat soluble vitamins, you actually get more in oils. Like you get vitamin E in fish oils. Fat soluble vitamins, they are more non-polar. And these include vitamins A, D, E, and K. So if you eat a lot of this, and since they're fat soluble, you know that fat and water, what ends up happening, things that are polar and non-polar, separate. Okay, so fat soluble vitamins, they don't just circulate in the blood because the blood is a watery environment. So they don't just circulate. Instead, you store them. Where do you think you store them? In fat cells. They don't circulate. They end up getting stored in fat cells. So vitamin A is a vitamin that can get, it comes from like your orangey kind of vegetables like carrots. So it's beta carotene is the one. So vitamin A, if you eat a couple of carrots, and really you only have to have like 6 or 8 and you have like 100% of the vitamin A that you need. So my kid, my younger child, when she was little, she was like notorious for like eating baby carrots like without any type of control. And so it was like, she would like go to the refrigerator, open the cabinet, and she would like pull out the bag of baby carrots, and then I would like turn around and they would be all gone. And she'd be like, those were delicious. So at one point, her palms turned orange. Okay? And that is because underneath you have subcutaneous fat. So in the palms, you don't have pigmentation, like you don't tan, your palms and soles of your feet don't tan. So there's no melanin to like cover that up. So the palms and soles of her feet were orangey looking because she'd been eating so much vitamin A, because it was actually stored in the subcutaneous fat up underneath the skin. I was like, you can't eat all of these. It's not good for you. You can see in the dark. Because vitamin A is one that is involved in like the receptors in the retina. And so it actually improves night vision. My father was like all about like eating his carrot like every other day. He would have like a carrot at lunchtime, and he'd be driving along. It'd be pitch dark. My mother would be like, I don't even know where we're going. He's like, I'm fine, I can see. And he like claimed, he was like, it's because I eat carrots all the time. Is the whole reason for it. But it doesn't circulate. It gets stored in fat cells. And in fact, high levels of the fat soluble vitamins can be toxic. So high levels of vitamin A, the reason why I was like, you shouldn't be eating that many baby carrots is because it can end up causing some like neurological stimulation. So you can end up with like, you know, like the itchy skin and like weird sensations and stuff because of it. So some of those ones, especially the polar ones, it doesn't matter. If you eat a ton of it, it's just going to get excreted. But the fat soluble vitamins, they don't circulate. They get stored in fat cells, and they can actually build up. They can actually build up. Come on, stop that. There. If you overdose on them. Vitamin K is an interesting one, though, because vitamin K is actually made by the bacteria in your gut. So it's made, so it's like, so non-absorbed material moves along, and bacteria take the precursor to vitamin K and make it, and then it gets absorbed. So it's actually a benefit of something that your GI tract actually does for you, is it makes vitamin K. Fat soluble and water soluble, polar and non-polar ones. So if you look at molecules, things that you look for an irregular arrangement of electronegatives like oxygen, nitrogen. So remember, like in sugars, and this one shows sucrose. So on top is glucose, on the bottom is fructose. But see all those alcohols? See where the water molecules can come in and interact with all those alcohols, the oxygen here? And this is the reason why you put table sugar in water, stir it up. It can interact with water and stay mixed. It won't separate back out. So having a small molecule and only one nitrogen, oxygen, fluorine, or chlorine, that's going to be polar if you only have one and it's little. But when you're looking at bigger molecules, having a lot of like alcohols on it or having a lot of nitrogens, those will all improve the solubility by making it more polar. Those fatty acids, though, cause all kinds of trouble. Fatty acids, they're polar and they're non-polar. And this one's really not as polar and non-polar as you want. There is a carboxylic acid side. That's the polar side. And then the long non-polar tail. So if you have just fatty acids, you've got both parts to it. They call that amphipathic. So amphi means both. So it's like both polar and non-polar. Soaps are like a fatty acid, except instead of just having a carboxylic acid, it has a salt. And so it looks a little different. Oh, I thought I had it here. So it has the long hydrocarbon, long carbon chain. But then on the end, you have the oxygen, and an oxygen that is actually an ion, a negatively charged oxygen on that end, and that's going to add to the polarness, the charged nature on that side. So this is really more like a salt, more of an ionic interaction. But then you got this long non-polar tail. So when you put this in water, the nonpolar molecule does not want, the nonpolar side does not want to interact with water, but the salt side can. The salt side is going to want to arrange itself so that it can interact with water. And that's where you make the micelle. So a micelle is the name for a sphere that is made when soap molecules get put in water. The polar heads all form the outside. The nonpolar tails all go to the inside. This is going to allow the soap molecules to interact with water and anything that's water soluble, but it's also going to create a region that's nonpolar, a region that can interact with any kind of greasy, oily type of molecules, which makes it a great way to be able to break up a stain. So another term they use for nonpolar is they'll call it hydrophobic, because a phobia is what? Fear. So the term phobia, like if you have a phobia, you're scared of something, like arachnophobia, you're scared of spiders. So hydrophobic means that it's water fearing, whereas hydrophilic, the term philic means loving. So hydrophilic wants to hang out with water, hydrophobic does not. So here is the micelle. And in lab, we kind of looked at them and talked about them twice. Once when we did the stain removing lab, once when we made soap itself. And so you can see out here the polar head. See how it wants to interact and has the ability to interact with water molecules surrounding it. So those micelles will dissolve completely. Remember, it made that soapy water cloudy. So you start off with warm water and then you added just one small scoop of soap flakes and stirred it, and immediately it got really cloudy. The soap completely dissolved. That is because there were millions of these floating in the water. But see these nonpolar tails all sticking to the interior? when you wash with soap, as soon as you, and if you just use soap, it doesn't work. You have to add water. You have to add water or you won't end up with any micelles. So like getting like a squirt of liquid soap, there's some water in your liquid soap, but you really need to add a little bit of water to try and get it to suds up to foam, and that helps you to create those micelles. So in this whole hand washing aspect, so you have billions of bacteria that are like all on your body surfaces, like you can't scrub them off, and they live there. A lot of the bacteria are called resident. Right? So if you're a resident, then you live there. Resident bacteria actually live in the layers of the outer parts of the skin, the epidermis. So they live in like those outer 15 layers. So they're all down kind of trenched in. When you wash your hands, you're really not going to get rid of these because you would have to just wash your hands for about an hour and a half or so before you'd really start getting a lot of the resident bacteria out. But the ones you're more concerned about, so resident, these are always there. And they're found deep in the skin surface. But if you wash your hands and you allow enough time to form micelles and mix on the surface, you're going to get to the bacteria that are on top. So where did the bacteria on top come from? They're not resident. What do they call them? Where did you get them? Because you did that. Because you went like this and you picked up the snot that somebody left on the countertop. When you went like this. And you can't see them because they're microbes, they're microscopic, but those bacteria are on the surface. Those bacteria, they're not down in 5-10 layers down. They're right on the surface, and those are the ones that the micelles are going to get off. Okay, so resident bacteria are always there. And your resident bacteria, it's perfectly fine. It's considered the healthy flora of your skin, but the transient. So when you think of something that's transient, what's that mean? Yeah, sometimes there, sometimes gone. Okay, transient bacteria are the bacteria that you pick up by touching other things. The bacteria you pick up when you handle patients. Okay, the bacteria you pick up when you handle specimens. So in doing any type of healthcare especially, so it could be anything from changing bedding, so you could pick up bacteria there, but they're right on the top. They haven't like gotten down. They don't live there like year round on your skin. They're just there like short term. And they're the ones that oftentimes will make your patient sick. These are the ones that may come from an infectious sample, that they may, even in changing beddings. So maybe the person like stooled themselves, or maybe that person had an abscess and it's draining. So all of that is potential, and those are all going to be transient. And so that's where the hand washing and making sure that you're doing it for 15 to 20 seconds, not just running water. Because if you run water, just like run water on your skin, what are you doing? All you're doing is just wetting the transients. You're really not. They have actually shown that like physical rubbing your hands, that helps loosen transients. Even using paper towels is better in removing transients than using the air dryer, because the air dryer just kind of dries them off. So any that didn't get washed off, they just then dry back up and they're still there. Whereas when you take a paper towel and you rub your hands, you actually remove more of the transients that way too. So there's a lot of like research that they're constantly doing, because working with patients, you're always worried about picking up transient bacteria and then taking it to the next room. And you don't see it, and you might be like, my hands are perfectly clean and this patient's perfectly healthy. But the bacteria of this patient might make this patient sick. This second patient may have a surgical incision. And so that's an open wound. And so you may transfer bacteria from one patient to another, and hand washing is like the number one way to stop that. Cool. Yeah. So all of those, yeah, your MRSAs, your VREs, yeah. And there are some, so there now is a Pseudomonas. There now is a Pseudomonas that no antibiotic kills it. And Pseudomonas is one that you see in the damp, wet, like open wounds, burns, that type of thing. So like that's a pretty nasty bacteria. Like it used to not. I mean, you used to find it in like water fountains and stuff because it lives like in damp areas. Wasn't that big of a thing, but there actually is now. They have had patients that have got contracted a Pseudomonas strain that is like completely antibiotic resistant. They can give you one, but it'll kill your kidneys. So that'll fix you. We can treat you for this infection, but it's going to shut your kidneys down on a permanent basis. So that's terrible. So transient is picked up. It is much more superficial. And so it doesn't stay there all the time. And that's one. Transient is really the one that hand washing can help. Really can get the transient ones that you're picking up. Because a hospital really is just a high concentration of germs. Okay? Because why do you go to a hospital? Because you're sick. Okay? So if you go to the hospital because you're sick, so did the guy next to you. So did the guy in the next room and the one down the hall too. And so therefore, that means that you've got a higher than normal percentage of weird transient bacteria that you wouldn't get in the grocery store. Well, I don't know, maybe a Walmart, but just in general, you wouldn't get at a grocery store, you wouldn't get in a schoolroom. Okay? And it's because you are purposely going into an environment where there's people that are sick. People that have higher microbe numbers. And so those transient bacteria are really the ones that hand washing comes about. That's the soap production one. So we talked about soap. All right, so that leads us to the last of those topics, and that is most like soap are the phospholipids. So phospholipids are a group of lipids that make up cell membranes. And they actually look a lot like a micelle, except instead of forming a single sphere, they form a double layer. So they have two long carbon chains, fatty acid chains, and they have a charged phosphate containing group that creates their polar region. So they end up, instead of having just one long tail, they have two. Very similar to a soap molecule. They're just a little bit bigger. This is that polar region, and this is your nonpolar region. But very similar to just the fatty acid types. And they actually have an image of chemical structure. And you can see the tails. Top one is a saturated fatty acid. Bottom one's a monounsaturated. See how it makes the bend in it. And then over on this side, so here you can see there's the ester. So those oxygens are going to help and add to the polar nature. Here's a phosphate, and that phosphate is charged. So that's more like a salt. Same thing here. Here's a nitrogen on this end, and that's got a positive charge. So also making it a lot more like a salt. Remember we said in terms of strength, ionic compounds have the strongest intermolecular force. So these like to cling together. So when you take a bunch of phospholipids, so this is what they call the cartoon version, with the heads and the two wiggly tails, when you put these together, they form a bilayer. So they form a bilayer where you have a layer of phospholipids and a layer of phospholipids below, and their tails all point inward. Like this. So these are all the polar heads with those charged parts, the oxygens, creating that polar part. And then here's all of the nonpolar tails. So instead of forming a sphere, it actually forms a layer. I think of it looking like a little sandwich. So the top and the bottom are the polar parts. And so out here, anything watery can interact with the polar heads. Anything like a salt can interact with the polar heads. Sugars can interact with those polar heads. That polar head is going to be water interactive. so they can interact. But this layer creates the barrier. Anything that's watery can't pass through. Anything that's salty can't pass through. Anything that's polar can't pass through. So sugars, boink, they bounce off. They can't just pass through the sandwich. They can't pass through the middle because it's oily. And you know oil and water don't mix. So anything that's water soluble can't freely pass through. And this makes this great cover. So this is a great cover on structures in the body, cells. This is what the cell membrane is based on. And it's going to mean that now you've got a cover that things just can't move freely in and out. Creates this oily barrier to anything water soluble inside or outside. So things can't move in, but things can't move out either, unless they're nonpolar. Oxygen's nonpolar, carbon dioxide's nonpolar. So oxygen molecules, they diffuse right through. They can actually pass through this barrier. They're a gas, but they're also nonpolar. So they don't have that barrier to them. Same thing with carbon dioxide. But anything sugary, anything watery, anything salty, those things aren't able to pass straight through. Instead, they have to have a transporter. So if you look at this one, and I can blow it up for a minute and then we'll blow it back. Here's the big one. So the green, all the little green, that's the phospholipid barrier. But then we've also got a couple of things that are anchored in here. So the phospholipid barrier really makes that cover. But in the phospholipid membrane are these blue structures. So the blue structures are proteins, and we'll talk about proteins in chapter 10. See the yellow ones? They look like jelly beans. See the yellow ones that are in there. So the yellow ones, those are actually cholesterol molecules. And those are really the three things that you find in a cell membrane. The phospholipids, they make up the bulk of the membrane, and they're really a barrier to anything that's polar, anything that's ionic. Those things cannot freely move through. Then you've got cholesterol. Cholesterol is the one that's like yellow. Cholesterol, we'll talk about cholesterol next time, because cholesterol helps to add stability to the membrane. Animals or plants? Which organisms make cholesterol? Animals, okay? Because we're warm-blooded. And so we need to make our cell membranes a little more rigid because we're at a higher body temperature. Trees don't have to do that. Okay, so trees don't make cholesterol. Peanuts don't make cholesterol. So when they say cholesterol free on a peanut butter jar, I'm like, never had cholesterol. They didn't remove it. No plants make cholesterol. And so that's the difference in it. But it just helps to make the membrane more stable. So this is like an animal cell membrane. The other one, though, is the proteins. So the proteins, if they are integral proteins go all the way through. So see this one? It goes like, you can see that the protein is on one side and the other side. So that's what integral means, kind of throughout. So it passes, it is present on both sides of the membrane. And what does that one look like? The one I underlined. You see how it's like this and has that little hole in the middle? Looks like a channel, doesn't it? Doesn't it look like a tunnel? Okay, that is how you can move molecules in and out. So we do need to pull glucose in. So there is an integral protein that will allow glucose to pass through that channel. It'll pass through that hole, but what has to be there? What has to be there in order for glucose to do that? Insulin. So there's actually like a little receptor that you have. So this one, see this one here? This one here These ones are only attached on one side or the other. So they call those peripheral. So if you're peripheral, that means you're sort of like around the edges. Instead of going all the way through, peripheral. A lot of times peripheral, these can be like anchors. They'll hold cells together. So one cell gets lashed to the other cell by a peripheral protein. Sometimes these are enzymes. Like in your GI tract, you have enzymes that hang out off of the surface of the small intestine. And as lactose goes by, chomp, it breaks it down. So they're enzymes that are like attached to the surface, and they only stick out on one side. So they're only on one side or the other side. They're not on both. So peripheral proteins. Integral, these can be things like channels. Peripheral proteins, a lot of times they help to anchor. But here's this. Where have we seen that? A carbohydrate group. Carbohydrate groups that may be attached to proteins. See how like this one, it looks like it's attached to it? When did we see that? Carbohydrate patterns. No. On a cell as an identifier. Blood typing, right? So those, see the little things, they look like little antlers, little limbs sticking out. This would be an example like with your ABO blood group typing. So the pattern of carbohydrates, notice they're only on the outside. Why would you have them on the inside? Nobody cares. They have to be on the outside because they're marking those white blood cells saying, we're here and we're part of us. Like, please don't destroy us because we're part of self. So they're only just on that one side. So those would all be part of that peripheral proteins. They're attached to, and if you look at it, I'll blow it up one more time. If you look, see like how the antler looks like it's sticking on like a little blue? And so that's a peripheral protein with carbohydrate groups on it. And so those are things like your ABO blood group types. They help to mark the cell as part of you. Mark it as self. Tells your white blood cells, don't attack and destroy me because I belong here. Okay, so those are really like the three main parts of a cell membrane. Phospholipids make the barrier. Cholesterol makes it more stable. Proteins allow things to go in and out or help to do specific functions for the membrane itself. All right, I'll quit there. I wanted to do one thing. I want you to see if you can do it. So I brought some. So to show you some surface tension. Oh, there's water everywhere. You can see if you can do it. So here is, I won't be able to get it. So here is a beaker of water. Here's your water bug, and there's your dipper. I want you to see if you can actually get your water bug to float on the surface of the water. Okay, that's your water bug. And then the other paper clip is your dipper. I don't care who does it. Did you do it? Okay, and then you can actually see, can you see the dents that it makes? So you want to take, so your water bug, you have to put it on the dipper, set it on there. It has to lay nice and flat. You have to put your flattened paper clip, is our water bug, and you're putting it on the bigger paper clip, which is the one to help dip it. You want it to be nice and flat. You want your water bug to be nice and flat. Fish it back out. Let's see. Lay it right up there. Nice and slow. Lower it down. Lower it down. Dropping? No. You have to spread your water bug out. So your water bug, like you pushed it together, it needs to have more, it needs to like cover more area.
Keep going down.
Nope, not like that.
Slowly.
I was trying to figure out how to do it. Okay.
That's your dipper.
Oh, you almost have.
It's
It's
spread
because
it
it's
out.
wet now. It's easier when it's dry.
And then see. Yeah, and you do have to dry it off if it gets wet. That does help too.
Yeah, dry.
But that's what, so surface tension, all those hydrogen bonds they're strong enough to hold the paper clip and keep it right on the top.
All right, here you go. Just
Nice and
round two.
slow.
Yeah.
Here we go
Take
through.
it.
And then see if you can get it out of the way. And then you can lift it up and out.
All right.
And can you see the dents?
Very, very slow.
Even though it's a paper clip, even though it's metal, like I always see water bugs, you're like, yeah, they don't weigh anything. That's a pretty heavy thing just for a paper clip. But the hydrogen bonds actually can support that paper clip and keep it from being able to break through. Did you get it? But if you shake it or move it, you'll break the hydrogen bonds and it'll drop to the bottom really fast. I should have brought soap because if you add soap, it breaks the surface tension and it'll drop into. That's all because of hydrogen bonding that it can do that. All right, so what we've got to do is we're just going to finish this little bit about cholesterol and the cell membrane. We have to talk about gases, and then we're going to go on to chapter 8 on Monday night, okay? So you do have time to do the pre-lecture for 8 if you haven't done it. Make sure that you get it done. Just go ahead and do 8 and 9 because they're both open now. The pre-lectures for 8 and 9.